saying that I see the pooling as working for all concerned in that region, in terms of if they have an interest in going forward to mine. That's my response.
I totally understand what the goal of this workshop and also what goal of the ISA. My concern is for sharing the data, it is a good idea to be the managing total place of the CCFG, and also your statement is I 100% agree. However, maybe some of contractors still concerning about that they opened their whole things the beginning of it. And what I mean is maybe the process of the sharing data should be something regulated or something making all contractors be understand. And that's what I'm saying. It's not I'm not object about doing that, but to process of the sharing data and also do some of the things we go forward to the development. And before then, we think need for the all agreement of everybody, every contractors sharing data. That's I little bit concerned.
I think one way to address some of those concerns, which I can understand, is that I think the sharing of data is about asking specific questions. So we're not going to ask for all of the data. We're going to ask for particular parts of the data that relate to a question that is of concern for everyone. And I think the other aspect of it is that we have to make sure that everybody's producing the same sorts of data so that we can address those questions. So that one group will not feel that they're putting all the investment into answering this question and another group aren't. And I think that's a perfectly reasonable attitude. I think we have to be open, but we have to make sure that we do get data from all of the areas we need it. And that's right across the CCSA. So all of the contractors have both the opportunity to do that, but also they will get the benefit if they're contributing to this.
Go ahead.
Sorry, I know we need to move on. I just wanted to make a point as a contractor who's been collecting data for SMS deposits for a number of years. The value of sharing everything that we have with contractors who might not have much to date is very well understood from our perspective for two reasons. One, obviously, if I have data about this little system here, it's of almost no value if I don't have any data about the rest of the area, and I think we've well understood that. So that does add a significant amount of value to our data. The second thing that adds value is that when we take our data and make it publicly available, and there's an opportunity for experts like those in this room to really interrogate it and ask questions that are difficult and really cause us to think about the robustness of it, it gains another value that we just simply can't get by holding that data to us. So if we have a report that's in our EIS or made available in some way, and that data has been made publicly available and scientists around the world have had an opportunity to interrogate it and make comment on it, then we look more robust, and it's a value for us as contractors. And I think that's an important thing to remember as well.
If I may, I have something to add to what my colleague was just saying. As one of the newcomers that is just starting with collecting data, just to add to the previous comment, I think all the contractors are well aware of the commitment they made towards the ISA. We all entered into a contract for exploration, which clearly entails that we will need to report to the ISA about environmental data, which is to be meant not to be public, to be meant public, but we report to the ISA. And it's up to the discretion of the ISA to make public whatever they can make public. And the sensitive information regarding commercial aspects will be kept confidential. So I don't see the issue, to be honest. Thank you.
Any other questions, comments? So we've pass to the second lecture, Lenaik Menot. He will be talking to us about the ecological context for the study of biodiversity of the macrofauna of the Clarion Clipperton Fracture Zone.
Thank you. Good morning, everybody. I'm going to reiterate some of the points that is made by Gordon from slightly different perspective. I'm going to consider two reasons why we are interested in studying the macrofauna in the CCZ. The first one is rather related to the obligation of the contractors to monitor the consequences of the activities, potentially the consequences of illegal mining. So this is the part of impact assessment, which needs guideline studies and which needs the development of indicators of impact assessment. Or can we quantify any impact? The second reason why we are interested in the macrofauna is rather related to the need for the authority to prevent any significant adverse impact of the environment. So mitigating the consequences of the mining could be through marine spatial planning, like the areas for particular environmental interest. And for that we need to know if these areas are representative of the CCZ, if there is connectivity among these areas or between these areas and the areas that are going to be mined. To achieve these two goals, there is a number of ecological, biological properties of the abyss that are quite challenging. First of all, the primary productivity in this area of the Pacific is quite low. So the food that is sustaining, that is providing to the seafloor is quite limited, and so densities are very low. Usually, when we are taking a sample of macrofauna, so we are using a box score, which is 50 centimeters per 50 centimeters, so a quarter of the square meter. And then we managed to collect on average 25 individual of macrofauna per crop. So densities are limited, but species richness is high. So if this is the same kind of example that was given by Gordon. If we look at the number of species according to the area that is sampled, we can sample up to 100 of species in 4 square meters. So on average, 400 specimens, 100 species. So on average, 4 specimens per species, not much. And in fact, many of these species are rare. So many of the species, we only get them in just one crop. If you look at the most abundant species, this is an example from Esler and Jumar. So they have taken 12 box scores. Out of these 12 box scores, they managed to identify 54 species of polypid, it was. Half of them were rare. And the most abundant one had about, so they managed to collect 30 to 35 specimens of the two most abundant ones in total, which means about 3 individuals per box score. And because of this long list of rare species, if you compare the species composition of your sample, the similarity in species composition is low. study station, they conclude the same for many, many species. And these has consequences. It has some consequences on the way we are going to monitor the impact of mining. So there are some kind of methodological issues, and also there are some concerns about some theoretical issues associated with the way we might try to mitigate the impact of illegal mining. And that's two questions or two issues that I'm going to address in this talk. The methodological issues is related to the power of the statistical tests that we are using. So it's just kind of short introduction to statistics. So the processes, the parameters that we are studying in nature, they do vary. The size, for example, the size of men and women is not the same for everybody, so there is a variation of the size. Let's say size of women and the size of men. It's the same for the population we are studying. Densities are changing. Okay? There is variation. We don't know the scale of variation of the population in nature. We try to approximate that by taking samples. So with samples, we can compute the mean, let's say the mean density of one population, and we compute the variance, the variance of the density of the population. And then we compare our two populations. And we want to know, is the difference I see between population 1 and population 2 means that I have two different kinds of population, or is it the same? Then it's the same. Could have taken 3 samples from this part of the distribution of densities and 3 samples from this part of the distribution, I will get this kind of graph, all belonging to the same population. So in order to know if these variations are true variation in nature or not, we are using statistical tests. Usually we have 2 hypotheses. The null hypothesis is this variation doesn't mean nothing, or the alternative one is these variations mean something. There is something going on. As we are doing this, we always have the risk to make an error. So let's imagine that we have here an area that has been impacted, and here a control area. We want to know if indeed this area has been impacted, so this difference is significant or not. So the first kind of error that we can make is that we decide that the null hypothesis is false, where in fact it's true. So we decide that there is an impact when in fact there is no impact. Okay. That's the first type of error that we can make. That's the one that we try to deal with in general. And the probability to make this impact is alpha, and by convention, alpha is 5%. So it's quite low probability. We want to make sure that if we conclude that there is an impact, really there is an impact. But there is another kind of error. And this error is when we conclude there is no impact, but in fact there is one. That's the kind of error that usually we don't deal with. In most cases, we just don't look at this kind of error. We just don't look at the probability of making this error. This is the error of type 2. And the probability of making this error is beta. And beta is giving you the power of your analysis. Okay? So, basically, this means that if you want to make sure that you will be able to conclude that there is an impact, you need to have a good power, so a low probability, low beta probability. And the beta probability is a function of variance, the effect size, so the scale of the impact, and the sample size. So I'm not going into details about how to compute power analysis. It's becoming tricky. Just to illustrate this, a kind of graph analysis, but simple. If you look at the precision of a density estimate, how good is my estimate of the mean density of the population? This can be computed as a function of the mean density, the variance, and the number of samples. And if we assume that our organisms are randomly distributed, then the variance equals the mean. And then we can compute the number of samples we need to get some level of precision as a function of the mean and the precision. And we know that if you want to detect a half decrease in density of a species, let's say, so there is an impact, this impact led to a half decrease in the density of one kind of sensitive species. So if you want to detect this half decrease in density, we need a precision of 20% on our mean density, so 0.2. And from this, we can compute the number of samples we need to detect this effect size using the prediction according to densities. Okay? Looks like that. The mean densities per sample, the number of samples. So, for the most advanced species in the CCZ, so about 3 individuals per sample, you will need about 10 box per area. 10 box per in the area that has been impacted, 10 box per in the control area. That's for the most prevalent species, 1 or 2 species. For most of the other species, you will need from 25 up to 1,000 box scores to be able to conclude if there is or not an impact with some level of accuracy. Okay. So that's the first issue. And the question we can ask from that is it realistic? Can we really achieve a sampling strategy that is good enough so that we can really detect an impact in the CCZ? If not, how can we overcome this problem? We can think about taking larger samples, taking more individual per sample. Like, for example, with a triple-helix sledge. The sledge, however, has some problems. It's not quantitative, and it integrates many scales of heterogeneity in the field. So this is something we have to address. The answer to that. The other way to overcome this problem of power is what we call taxonomic efficiency. And taxonomic efficiency is which level of taxonomic resolution do you need to detect an impact? And in shallow seas, it has been shown that species level maybe is not needed. You can work at family level sometimes. So from that, we might ask, do we need species identification with a big question mark? We don't know. This is something something we have to address. It's something we have to test. To test it, first of all, we need species identification. So, until we have solved this problem, or address this issue, we need species identification. And then, considering the second kind of issue we have in the CCZ, it's related to the coexistence of species and the maintenance of diversity in an ecosystem. Looks like a very basic question, but it has some consequences on preservation. And it comes to a kind of important paradigm we have in ecology, which is called competitive exclusion, which says that two species that are using the same resource in the same place cannot coexist. There is one species which is better than the other, and the good one is excluding the bad one. Okay. This has been shown experimentally, and there are some theories and mathematics around that as well. And this idea lead to the definition of the ecological niche by Evin and Chilson. So the ecological niche is kind of space. The space is defined by all resources a species is using. It's ecological niche. Two species cannot share the same ecological niche. Okay. In theory, species must have different ecological niche to coexist in the same area environment. And in some cases, it's pretty difficult to fit this theory with what we observe in nature, especially in areas where there is not that much heterogeneity, not that much resources available, but still a lot of species, like in the deep sea. So the question is, how can hundreds of species coexist in the same place with just one or two different resources? Very basic question for science. And to explain how it can work, different kind of models have been developed, which can be separated into basically four major processes: patch dynamic, species sorting, mass effect, virtual models. What does it mean? Patch dynamic. So in patch dynamic, you assume that you have the same environment everywhere, but some species are good competitors,